So we're going to go ahead and go over these. So the first question here, we had a scientist that determined a specific DNA molecule consists of 30% adenine, and you were asked to calculate the percentage of cytosine present in the DNA molecule. What did y'all come up with? 20%. That is correct. So 20% cytosine. If you had trouble figuring that out, what pairs with A, y'all? T. So how much T should we have in this molecule? 30%, okay? So we know that adenine pairs with thymine, and so together they make up 60% of the molecule. That means the remaining percentage should be made up of C and G, and since those base pair together, then half and half, that gives us 20% cytosine. Any questions about that first example? The second example, a scientist determines that a specific RNA molecule consists of 20% guanine. Calculate the percentage of thymine in this RNA molecule. It is 0%. It is a trick question because what does RNA not have? Thymine. RNA does not have thymine, so it has what? Uracil. Absolutely, it has uracil. So RNA uses uracil. That U. Okay. There was an AP Bio question a couple years ago where they asked, it was like a multiple choice question kind of like this, and they asked, how much thymine is in an RNA molecule that consists of 20% guanine, and you best believe they had 30% as an option on there because they wanted you to think that there was thymine in that RNA. So just don't let them trick you. Make sure you're looking at the fact that this is an RNA molecule and that there's some different bases there. Any questions about that? That one was fairly straightforward except for the fact that it was a trick question. Okay, the last example is actually two questions. The first part asks, what does the DNA of all organisms on Earth have in common? What do we think, y'all? To an extent, it does have to do with the base pairs. Yes, sorry. They will all have adenine, guanine, cytosine, and in the DNA specifically, put it there. Yeah. Oh, yeah. Adenine, cytosine, thymine, and guanine. So the ratio of the base pair, the ratio of the A and the T should be the same, and the ratio of the G and the C should be the same. But the actual percentages of A's, C's, G's, and T's may vary by species potentially. But yes, they do all use the same 4 base pairs. So if y'all want to write all organisms, use the same 4 base pairs. Or if you just want to write that they all have, A, C, G, and T, then awesome, that works too. Okay? And what does that suggest about life on Earth? The order of the base pairs will determine the different diversity we see between species. That's going to be different sequences will result in different proteins being coded for. Is that what you were going to say? Yeah. The common ancestry? Yeah. Yeah, no, it's good, yeah. Yes, common ancestry. So this does suggest that all organisms seem to be descended from a common ancestor. So that's our theory of evolution that kind of underpins all of biology. Okay, so all organisms seem to be descended from a common ancestor. Okay. Any lingering questions there before we jump into DNA replication today? Okay, so DNA replication. As we go through DNA replication today, this is going to hopefully just kind of be a review, maybe clarify some things that you were confused about in first year bio, because DNA replication does confuse people sometimes with all the different enzymes and such. I have a lot of different pictures to help us kind of talk through what's happening and what each of the enzymes that we need to know are doing during the DNA replication process. I also am going to show y'all some animations that are 3D models of DNA replication. Those will be linked on Canvas, so if you're watching the video to get caught up for some reason, then you can watch the video on Canvas later. And we will be showing those in class after we talk about the basics of DNA replication. So DNA replication begins at special sites called origins of replication. So that's the first thing you're going to write here, origins of replication. This is where the DNA strands are separated. As the DNA strands are separated out, what opens up is something called a replication bubble. So it looks like it's kind of like this little bubble in between the two strands. That is your replication bubble. And so in a prokaryotic cell, such as an E. coli cell, E. coli is bacteria, and bacteria are prokaryotic, you may recall that they have circular DNA. They've got circular DNA molecules. And so they'll have a little replication bubble open up in their circular DNA molecule, and the DNA will replicate out to both sides until we come essentially full circle and end up with two daughter strands. In eukaryotes, it's a little bit different. Yes, Ali?
So is the bubble where the two strands separate?
Yes. Yeah, so that where they separate, that is the bubble right there. And then at the end of that bubble, we have something else. It's called a replication fork. So the bubbles, there's typically just like kind of one in your prokaryotic cell. In a eukaryotic cell, you may have many or even, thousands of origins of replication in a very long linear chromosome. The DNA strands, as they replicate, they will eventually, collide with each other, and a special enzyme will make sure that everything is all glued together there at the end. So replication, we say, is going to proceed in both directions. Both directions from each origin until the entire DNA molecule gets copied. So we can see these new daughter strands are going to be created, and because DNA has that complementary base pairing system, a parent strand acts as a template for a daughter strand because of those base pairing rules. It's pretty efficient. At the end of our replication bubbles, when those strands separate out initially, we will see something form called a replication fork. That is that little Y-shaped region that you see circled here. So you can write at the end of each replication bubble, there's a replication fork. That's where the new DNA strands are elongating out towards, well, one of the strands is elongating towards the replication fork, one of them is technically elongating away from. We will talk about that on the next page. And there's a lot of different enzymes that facilitate the replication of DNA. Some of the enzymes College Board wants you to know for the AP exam, and so I'm going to star them on our notes so that way you know which ones to memorize for both your unit test as well as the AP exam come May. The first enzyme we're going to talk about is one that you need to memorize, and it is the enzyme that untwists the double helix. Do you remember what that enzyme is, y'all? Helicase. Yeah, helicase. So helicase, it's got that ace ending. It's an enzyme. And then it starts with basically helix, essentially. This is the enzyme that untwists the double helix. And this is one of the enzymes that College Board wants you to know by name and function. So I do want you to know that helicase untwists the double helix. So that is what is helping to open up a replication bubble, and it is going to be traveling down and pushing apart the strands in that replication fork area. As the DNA strands are separated out, as they get pushed apart, they do kind of have a tendency to want to snap back together. So there are some special proteins that will actually bind to the DNA strands to help keep them separate. You don't have to memorize the names of these, but they do show up in a lot of images that concern DNA replication. So I wanted to draw your attention to the single-strand binding protein. So the single-strand binding proteins help to stabilize single-stranded DNA, keep that those two parent strands separate so we can start to eventually synthesize a daughter strand, or two daughter strands. So we've got helicase and single-strand binding proteins. there's another enzyme that helps relieve some of the tension in the double helix as helicase untwists the double helix. Have any of y'all ever tried to like separate like a string that's like, oh yeah, Twizzlers, that's a good example too. Like Twizzlers, or this really happens a lot with I feel like string. If you've ever done any sewing, or you've tried to separate out the individual pieces of yarn, separate those out. What happens if you like pull it? It'll kind of ravel back up, and then at the end, like as you try to pull apart those strings, it gets like really bunched up and tangled too, which is really annoying. So you have to like stabilize the other end, and it's like a whole production trying to separate out the string. That same kind of thing, that same kind of concept happens with DNA too. Helicase, as it untwists the double helix, if we didn't have this other enzyme helping to kind of break and swivel and rejoin DNA strands a little bit down further in the strand would actually get really, really tangled up, and it would be a big problem. So there's this special enzyme called topoisomerase. Kind of a weird name, but topoisomerase will relieve the strain of the twisting of the double helix by, again, breaking, swiveling, rejoining DNA strands as needed as the helicase unwinds it. Yes, ma'am? This is also one of the enzymes you have to memorize. So go ahead and put a star next to it, yes. This is enzyme number 2. There's 5 enzymes that College Board lists by name that they want you to know the name and function of. This is number 2. So after we have unzipped our DNA, it is now, potentially going to be able to be replicated so long as all of our other enzymes are present. DNA polymerase specifically is what is going to help build the daughter strand, but DNA polymerases require a primer in order to start adding DNA nucleotides. So before the DNA polymerase comes in, a short RNA primer is going to be synthesized by a special enzyme. Does anybody remember this name? It is primase, yes. Primase. So synthesized by primase. The primase will start that RNA chain. It's going to add some RNA nucleotides. This is an RNA primer, so made of RNA. This is not an enzyme you have to specifically memorize, but it is an important enzyme, so I do like to mention it here. And so as this RNA primase is adding the RNA nucleotides, it is doing so by using the parental DNA strand as a template. So those base pairing rules are going to determine what RNA nucleotides get laid down. The primase is going to look at that parent strand. If there's an A, it's going to add a U. If there's a G, it's going to add a C, and so on and so forth. And so on the next page, you can see some of these enzymes on the image. I couldn't fit that image onto this page as well as this other replication fork image, so that's why they're separate on separate pages. But if you flip over here, you will see some of the players we've been talking about. We have helicase here. Helicase is, like we said, unzipping the DNA. We have topoisomerase. This is hanging out down here, helping to prevent the DNA strand from getting all bunched up and tangled. We have our single-strand binding proteins, helping to keep those strands separate, so that way the primase can come in and lay down a primer. And what's really important here about this primase is it is going to lay down some primers so that DNA polymerase can come in and start adding bases to a 3' end, okay? This short RNA primer, which is, it's only like 5 to 10 nucleotides long. It's really not long at all. It has this free 3' end that serves as the starting point for DNA polymerase. Once this primer has been laid down, DNA polymerase can come in and start adding DNA bases in this direction to this free 3' end. So we say, oh, yes, ma'am.
Does the topoisomerase make it so it's not curly, it's just straight?
Not sure if it, so the question was, does topoisomerase make the DNA so it's not curly, but so it's just straight? I'm not actually sure, actually. I want to say, no, I don't actually know the answer to that, so I'm not going to even try to answer that. But the DNA, that does bring up a good point. The DNA in this image does look very flat. It doesn't look like it's helical in nature. Doesn't have that double helix structure. That's just to kind of make it easier for you to see the base pairs here in that sense. But I would have to look that up for you to see if topoisomerase, if where it's at, it's helping to make it straight instead of helical at that point. But I don't know the answer. That's a good question. Yeah. Any other questions, y'all? Okay. I will try to remember to look that up. Okay, so once we have that primer laid down, though, we are able to have our DNA polymerases come in and do their thing. So I want you to write that enzymes called DNA polymerases will catalyze, they help facilitate the synthesis of new DNA. And so when the DNA polymerases come in, they are going to add a nucleotide to the growing strand. We're going to be building this polymer, this DNA molecule, from all these nucleotides. And you'll notice that there's some phosphate groups hanging out on here. You don't need to know specifically that there's 3 phosphate groups on this incoming nucleotide and that 2 leave. But what is happening chemically here is that 2 of these phosphate groups will pop off, and this remaining phosphate group will form a covalent bond with the sugar molecule here, or the sugar piece of this backbone here. And that is going to happen via a synthesis reaction, specifically dehydration synthesis. So as each new monomer joins, is added to the growing DNA strand, a dehydration reaction is occurring. The other way we can describe a dehydration reaction is dehydration synthesis. This is way back in Unit 1 where we talked about monomers and polymers. Building a polymer via dehydration synthesis. A water molecule is going to leave in the process of this new covalent bond being formed. And specifically, DNA polymerase can only add nucleotides to the free 3' end of a sugar phosphate backbone. And what I mean by 3' end, I'm going to go over to another image to help explain this better, is the 3' end is referring to the fact that there is a OH, a hydroxyl group, that is coming off of the number 3 carbon on that sugar. So we had mentioned, I think earlier this year, but it's been a while, that organic chemists, they will number carbons in molecules in order to, talk about specific carbons within a molecule. So this carbon right here in this deoxyribose sugar is what we would call carbon number 3. This is carbon 1, 2, 3, 4, and 5. There's 5 carbons here. And this carbon number 3 has an OH group coming off of it. This is why we call this the 3' end, because there's an OH coming off, a hydroxyl coming off of this number 3 carbon. Up here, this is called the 5' end, because carbon number 5, the free portion of this sugar phosphate backbone, is a phosphate group that is attached to carbon number 5. So that's where the 5' end gets its name. Now, we can only add a we can only create a growing chain adding to this free 3' end because we are going to have a dehydration reaction occur here. And so this hydroxyl group is going to be involved in the dehydration reaction that occurs when we add a new nucleotide to this chain. And so that is why DNA polymerase can only add in that direction. So if we go back over here, you will notice that free 3' end is where we are adding our growing nucleotide 2, our growing nucleotide chain 2. Okay, so that's what we're going to talk about down here with anti-parallel elongation. Because DNA is anti-parallel in nature, one of the strands is going to run 5' to 3', and if we go look at the other strand on the other side, it's running 3' to 5'. Because of the anti-parallel nature of DNA, the replication is going to get a little bit wonky depending on what side of the origin of replication you're on. Because DNA polymerases add nucleotides only to the free 3' end, they add nucleotides only to the free 3' end of a growing strand, The new DNA strand, we say, only elongates in the 5' to 3' direction. And this is really important, so I'm going to put some little stars here. The DNA polymerase only adds to a free 3' end, which means that the strand only elongates in the 5' to 3' direction. So here, for instance, is a 3' end. This is our free 3' end. Okay. DNA nucleotide bases are only added to this free 3' end. Okay. And so as we build this chain, this new daughter strand, we're adding to that free 3' end, and we're growing the strand in this direction. We're moving in this direction. This is the 5' to 3' direction. So this is why we say the strand grows in the 5' to 3' direction while we add nucleotides to the free 3' end. Yes, did you have a question, Valerie?
I figured it out. Thank you.
You figured it out? Okay. So that is our rule for DNA polymerases. They can only go 5' to 3, build a strand 5' to 3 by adding to that free 3' end. And so what that ends up doing is it ends up creating something called the leading strand and the lagging strand, depending on whether or not we are growing the new strand towards an opening replication fork or away from a replication fork, depending on, again, what side of the origin of replication you're on. A leading strand, you'll see, is synthesized continuously, while a lagging strand has to be synthesized in fragments. And so to make this make a little bit more sense with this whole 5', 3' thing, I want to go ahead and label these parent strands with 5' and 3'. Okay, so I'm going to make this side of this parent strand the 5' end and this side the 3' end. That would mean that on this side, this other parent strand would have to be the 3' end, and this would have to be the 5' end. And so if we have a parent strand right here, this parent strand running (5' to 3'), the daughter strand that we are synthesizing is going to have to be 5' to 3' in this direction, okay, just like we had that complementary parent strand over here. So as replication is happening, when this origin of replication opens up, this little bubble, initially as it opens up, okay, we have some space open up, we can lay down some primers, so that way we can start to synthesize some daughter strand, a daughter strand of DNA. Here, because as this replication bubble opens up more and more and more, it's just opening up more space, more parent strand for us to use as a template. This strand going in the 5' to 3' direction like this is going to be able to continually synthesize itself. So this strand is continually synthesized. Yes, ma'am?
So the pink dot is, the primer actually goes like on the parent strand, right?
Yes. The primer is going to be complementarily base paired to bases on that parent strand, and then the DNA polymerase is going to use that primer as kind of like a docking point to get established and start adding DNA bases to. Yes, that's right. On this strand right here? This right here, this would be, if we're looking at like this would be the 5' end, and this would be the 3' end of this daughter strand. Yeah, so this would be the 5' end of this daughter strand, and this would be the 3' end. If you imagine, because if we're looking at the two parent strands, if we initially just have our two parent strands, and I'll grab another piece of, before we open up this replication bubble, we started out with parent strands. One of the strands ran 5' to 3', the other strand would run 3' to 5' like this. If we open up that replication bubble and we start putting in some daughter strands, then those daughter strands are then going to have to also run anti-parallel to the parent strand. So this daughter strand, if we're just looking at like this right here, this daughter strand would have to run anti-parallel, so this would be the 5' end, and this would be the 3' end. Over here, on this one, this would be the 5' end, This would be the 5' end, and this would be the 3' end of that daughter strand. Does that make more sense? So depending on the way you're looking at it, this parent strand, if you're starting right here and you're looking at this parent strand, you could say that this parent strand is 3' to 5'. Or if you're talking about it from this direction, you would talk about 5' to 3'. I just like to say 5' to 3' a lot because DNA elongates in the 5' to 3' direction. And so the more I can say 5' to 3', the more I hope that it sticks in your head that it's running 5' to 3'. But these strands do actually run anti-parallel to one another. So you could say that this strand right here is going 3' to 5' in this direction, while this one is going 5' to 3' in this direction. Does that make more sense? Depends on how you're looking at it, and also specifically like if this is just like a kind of generic drawing right here. I didn't actually show that there's like a phosphate group coming off of the sugar phosphate backbone free at the end here, which is what a 5' end has, whereas a 3' end has the little OH, the little hydroxyl group. So we're just kind of assigning some numbers just to show this idea of anti-parallel structure. Does that make more sense? Yeah? Okay, cool. And do you have another question, Valerie?
So the primate sticks on there, right?
Mm-hmm.
And it moves from 5' to 3'
Yes.
for the new daughter genetic material.
Yes.
Then the polymerase comes after it and moves
Yes.
5' to 3'.
Yes. So after the primer has been laid down, these primers have been laid down, the polymerase now has a place to attach and lay down the DNA-based pairs.
So once the primer is on there, it doesn't slide down the new.
Once the primase will then dissociate from the strand. It will come off of the strand, and then the polymerase will jump onto the strand to take over. Yes. And then eventually these primers are also going to be taken out by a special enzyme that we don't have to know the name of, but an exonuclease will come in and take out those primers, and then another DNA polymerase will come in and replace everything, so that way everything is eventually all DNA. But right here, I wanted to point out that leading strand is going to be synthesized towards the fork continuously. So I want you to write that along one template strand of the DNA polymerase synthesizes a leading strand. The leading strand, again, is that strand that is synthesized continuously. It can be synthesized continuously because it's going 5' to 3' in the direction of the replication fork. As more and more of that parent strand becomes available, as that bubble increases in size, we are able to the DNA polymerase is able to just keep running along that parent strand and generating a daughter strand. That is not the case for our lagging strand. The other new strand, which we call the lagging strand, that has to be synthesized in pieces, in fragments. It is working in the direction away from the replication fork, so every time a little bit more of that replication fork opens up, it widens a little bit more. We now have more of our parent strand becoming available. So imagine initially when this is closed, this is, two strands together, we open a little bubble, now it's open a little bit. We can lay down a primer. We lay down that primer, and then we can synthesize some of our strand here, okay? Again, we open up a little more of our bubble, and then we lay down another primer, synthesize a little bit more of our strand. Oh, no. Always the lights have to go off at some point. Okay. And then same thing. It would just continue on and on until we did what we needed to do. This will make a little bit more sense, I think, once we watch the video, because sometimes it just makes more sense if you can see this actually happening in action. But I did want to just finish filling out our blanks here before we watch those videos. The lagging strand, since it is synthesized in fragments, we do actually have special names for those fragments. Do y'all remember what those are from first year bio? Yeah, they are. They're Okazaki fragments. Yeah. So the Okazaki fragments, they are named after, I believe it was a Japanese scientist who discovered these fragments, and so they ended up getting to name them. And these fragments are eventually going to be joined together by another enzyme that you need to know. Does anybody remember the enzyme that joins the Okazaki fragments together? Starts with an L. Yeah, it's ligase. Very good. DNA ligase. So DNA ligase is going to join the Okazaki fragments together at the end, and I'm going to go ahead and star DNA ligase because that's one of the enzymes that College Board wants you to really know. So at this point, I'm going to go ahead and pause the video, and I'm going to show y'all in class the videos of the DNA replication, and then we'll get back to our... Any other questions about leading versus lagging strand now that we're done with those videos? Okay. Then on the next page, you will see a image that summarizes all of the different proteins and enzymes we were just talking about, the leading strand, the lagging strand, so on and so forth, as well as a little chart summarizing the different enzymes. So the proteins and, specifically whether or not they're proteins or enzymes that participate in DNA replication are going to make this really large unit or complex that we refer to as the DNA replication machine. And the 4 enzymes that you need to be aware of that are part of the DNA replication machine, I'm going to come over in our little table and star them. We have all of the different enzymes kind of summarized here. So here's that DNA polymerase 1 and the DNA polymerase 2. You just need to know DNA polymerase in general. And so just DNA polymerase, topoisomerase, helicase, and DNA ligase are the enzymes you are responsible for memorizing the name and function of. And then you, of course, want to know in general how DNA replication works. Yes?
So those two, do we have to know both of those?
Both of these DNA polymerases, no. College Board typically doesn't ask you to differentiate. They don't typically differentiate even in their word problems, or sometimes they'll give you some more information and they'll call DNA polymerase 3, but you just need to know DNA polymerase as a general enzyme. Yeah. Okay. So those are your 4 enzymes that you want to know. The last 2 little things we're going to talk about before we get into the overview of transcription and translation is proofreading and repairing DNA and packaging DNA. So DNA polymerases are actually able to proofread newly made DNA and can replace incorrect nucleotides. So sometimes errors are made during DNA replication. The wrong base ends up being put down. It's not properly complementarily paired. And so the proofreading system can kick in. So if we, for instance, lay down the wrong base, if we put like a G, try to match a G with a T when we should have put an A down, then something called mismatch repair might happen. And in mismatch repair, the section of DNA that contains that incorrectly paired base will be taken out, and the DNA will be replaced with base pairs that are correctly base paired with the parent strand bases. Yes. So that is one way that the body can proofread and repair its DNA, guard itself sort of against errors that may happen during replication. Errors to DNA or mutations to DNA are not just going to come about during the process of replication, though. There are certain environmental factors that can cause DNA to become damaged. Certain chemicals or things like cigarette smoke and x-rays can damage DNA. Another really big one is UV radiation. A really common way that DNA gets damaged is via UV radiation, and this is why people maybe, parents may be on you about wearing sunscreen perhaps, because if you wear sunscreen, that helps prevent, some of those UV rays from reaching your skin and then your DNA. What happens when UV rays interact with DNA is that sometimes they can do something called, they can form something called a thymine dimer. The energy from UV rays can cause thymines that are next to each other in a DNA molecule to dimerize, to covalently bond to each other, rather than continue to interact via hydrogen bonding with the A's across from that. When they form this thymine dimer, that causes the DNA to kind of bulge out a bit. That can cause problems during DNA replication. It can cause problems during transcription, so on and so forth. And so if the cell catches that there's this thymine dimer, it might start to do nucleotide excision repair. It might cut out this piece of damaged DNA and replace it with new undamaged DNA to prevent complications from the formation of that thymine dimer. So you can write here that in nucleotide excision repair, a nuclease will cut out and replace damaged patches of DNA. I don't need y'all to differentiate between mismatch repair or nucleotide excision repair. I just wanted you to be aware that the body does have proofreading and repairing DNA mechanisms that it can use, and the error rate after this proofreading is done is low, but it is not zero. So sometimes things are missed. If the sequence change becomes permanent, if this mutation is not caught and the sequence change becomes permanent, like this for example, that could be passed down to future generations if that mutation was in a germline cell, aka a cell that produces gametes. If the mutation is just in a somatic cell, a cell that does not produce gametes, then that mutation will not be passed down to future generations, but it could potentially cause problems in that specific cell if that cell is not able to make certain proteins anymore because of a changed sequence, perhaps. So that's how certain things like skin cancer can develop, for instance. So that's what we have for proofreading and repairing DNA. Does anybody have any questions there? Then the last little thing I wanted to talk about was DNA packaging. In eukaryotic cells, DNA is packaged together with proteins called histones into something called chromatin. So if you have a complex of DNA and proteins in our eukaryotic cell, we call that chromatin. The way that all of our DNA actually fits into our cells is via a pretty elaborate packing system. The packing system of chromatin is, the way that chromatin can be packed is it can be packed very, very, very tightly together, or it can be packed much more loosely together. And that will actually affect gene expression. And we'll talk about that a lot more later this unit. But right now I want you to know that proteins called histones are part of this chromatin complex, and we wrap the DNA around the histones to package it, essentially. The unfolded chromatin that is very not tightly packaged is just the DNA around a couple of histones, and it looks a little bit like beads on a string. Like if you look at this down here, it's almost like there's little beads on a piece of string or something. And these little beads, as we might, refer to them, are what we call nucleosomes. A nucleosome is a segment of chromatin made up of some histones, some DNA wrapped around some histones. That's the basic unit of DNA packaging, and these are really, really important in gene expression, in regulating gene expression. Chromatin, as we talked about last unit briefly, does undergo some packaging changes during the cell cycle. The cell will, condense the chromatin down really, really tightly, condense it and pack it really, really tightly during mitosis. So chromosomes look like this during mitosis. We mentioned that when we went over the cell cycle. So a lot of people, when they think of a chromosome, they think of the stereotypical image of a chromosome. This is what chromosomes look like during mitosis. They don't look like that most of the time during the rest of interphase. There are certain sections of chromosomes that are a little bit more tightly packaged during interphase, but the whole entire chromosome you're typically not going to see that during interphase. It's just going to be during mitosis. And so, yes. And so we have two different types of chromatin that I want you to be aware of at this point. The loosely packed chromatin. The loosely packed chromatin is what we call euchromatin. And the tightly or highly condensed chromatin is heterochromatin. Depending on how tightly or how loosely packed chromatin is, can affect whether or not a gene gets expressed. You can think of this as like tightly packaged DNA is like, an object that is in a box at the bottom of like 3 other boxes in your closet. It's very, very hard to get to. It's very, very hard to express. The euchromatin is like our loosely packaged DNA. That's like, okay, that's just, sitting on the shelf in your closet. You can go and grab that real quick, express that information. So depending on how tightly packed our chromatin is, that will affect gene expression, which we will see a lot later in this unit when we start to talk about things like epigenetics. If your DNA is accessible, the gene is potentially active. We're able to get to that DNA, we're able to transcribe, get some information from that, and potentially make a protein out of it. Now, if the DNA is very, very tightly packed, very inaccessible, hidden away, folded away, tightly clumped, then a lot of times that gene is going to be inactive. It's hard for us to get to that gene and actually express information from that specific section of genetic code. So again, we'll talk more about that when we cover epigenetics a little bit later this unit, but that is the basics of DNA packaging. I wanted you to know that euchromatin is loosely packaged DNA, heterochromatin is tightly packaged, and changing the packaging can affect gene expression, which we're about to get into. Yeah, so we are going to talk, we're going to go through like 3 or 4 more pages. Yeah, I know. The notes this unit are a lot. So fun. Okay, so the next couple of pages we're going to overview transcription and translation, and then we will finish up transcription and RNA processing next time and then get into translation and mutation. So what I want us to be aware of today is the general flow of genetic information, the direction in which information flows, which is what we call the central dogma of biology. You may remember this from first-year biology. The flow of genetic information is DNA to RNA to protein. DNA to RNA to protein. And the actual information content of genes that is in the specific sequences of nucleotides. The order of the base pairs is the information encoded in the genes. Those are the instructions to make things like proteins. So the information content of genes is in the specific sequences of nucleotides. The DNA is what leads to the specific traits we observe in an organism, because the DNA is going to determine which proteins get made. Whatever instructions we have determine what proteins we can make. And so in other words, we say that proteins are going to be the link between genotype and phenotype. So whatever you have in the genotype, in the DNA, the sequence, is going to ultimately determine the phenotype that we see, the physical characteristics, the physical traits of an organism. And when we talk about gene expression, which is the topic of this unit, gene expression is referring to the process by which DNA directs protein synthesis. That includes the 2 really important stages that link the DNA and RNA and the RNA and protein, which are transcription and translation. And there were a lot of experiments done to explore the relationship between genes and proteins, this idea that genes determine what kinds of proteins get made. This idea was floated early on by a British physician in the early 1900s. His name was Archibald Garrod. He first suggested, or he's the first one we know of that suggested that genes dictate phenotypes. He thought genes dictated phenotypes specifically through enzymes that catalyze specific chemical reactions. So his thinking was that the physical characteristics of an organism, or of a person in his case, because he was treating people, okay? So the physical characteristics of a person were dictated by enzymes that that person had or did not have, enzymes that were functional or not functional in that person. He thought that symptoms of inherited diseases reflected an individual's inability to synthesize a certain functional enzyme. So, for example, people with the disease alkaptonuria, they do not have an enzyme that breaks down alkaptin. Most people have an enzyme that breaks down alkaptin. People with alkaptonuria do not. That results in the symptom of black urine. Okay, so when you can't break this down, you end up with this symptom of black urine. Okay? So he thought that all these genetic diseases that he was seeing were the result of the fact that somebody was lacking some kind of enzyme in some sort of metabolic pathway that was going to result in the symptoms that he saw in their disease. So we learned previously that there's many different pathways that cells use to make and break down molecules. Those are metabolic pathways. We spent a lot of time talking about the metabolic pathways of cellular respiration and photosynthesis. There's many other metabolic pathways in the cell besides cellular respiration and photosynthesis, though. And those can be implicated in disease as well. So to try and explore this idea of the gene and protein relationship, there were a couple of scientists that did some experiments. We had the scientists Beadle and Tatum, as well as Serb and Horowitz. They worked with this bread mold species, Neurospora crassa. What they did is they exposed this bread mold to X-rays to try and create mutants, because the only way you could really create mutants back in the day was just expose things to radiation and hope we created some mutants. So they did that. They subjected some bread mold to X-rays. They created some mutant bread mold strains. And they hypothesized that in each of the nutritional mutants they created, the gene for the enzyme that synthesizes a particular nutrient had been disabled. And so that was going to be responsible for the phenotype they were seeing, this lack of this enzyme in these different nutritional mutants. The results of their experiments helped provide support for something called the one gene, one enzyme hypothesis, which is the idea that the function of a gene is to dictate the production of a specific enzyme. There's a really bad H there. Sorry, guys. There we go. A little bit clearer. And so before you flip to the next page, I also want to point out one other thing here. I'm not really concerned with you deciphering this image over here so much as just getting the idea that they used X-rays to induce mutations. And I also wanted to point out the control that they used. They used wild-type cells as their control. We talked about wild-type strains or wild-type cells very, very briefly at the beginning of the year, way back in Unit 1. I mentioned that wild types are usually really good controls. If you ever see a wild type strain or a wild type cell group being used in an experiment, most of the time that's going to be your control. And the wild type, all that means, as a reminder, because we haven't talked about this in a while, wild type is just a variation of an organism Specifically, it's the variation of an organism that is found most commonly in nature. So I'm going to write, it's the variation of an organism most common in nature. They make very good controls. Because typically, when you're doing an experiment, you're looking at like mutants or something that has had a gene knocked out potentially, so you can try and like see what that gene was doing if we get rid of it. So that's a wild type. They're great controls. Is anyone still using what I have on this page? Okay. And then on the next page, we're going to kind of decipher this experiment as quickly as we can. So expanding on Beadle and Tatum's work, these two other scientists, Serb and Horowitz, they identified 3 classes of arginine-deficient mutants. These were mutants that were unable to make arginine. That's what arginine-deficient mutants means. And so each of these mutants that they looked at ended up lacking a specific enzyme that was necessary for synthesizing arginine. So in these organisms they were studying, there was this arginine synthesizing pathway. This is the metabolic pathway that they were looking at. So I'm going to make a little note that this is the arginine synthesizing pathway. You can see that an organism that has genes that are, present and not mutated can code for enzyme A, B, and C. Enzyme A allows an organism to make ornithine from a precursor molecule. Enzyme B allows them to make citrulline from ornithine, and enzyme C allows them to make arginine from citrulline. And the cells that they were working with specifically needed arginine to live. So I'm going to draw a little arrow there, and I'm going to say, these cells they were working with, that's my little abbreviation for with, needed arginine to survive. And so if they didn't have arginine or they weren't able to make arginine, then they would not grow in the growth medium. So they had 4 different strains of the Neurospora crassa. They had the wild type, which probably served as what in this experiment, y'all? The control, exactly. So their wild type served as their control. This was a good control because the wild type had all the enzymes. They had all the enzymes needed to synthesize arginine. That's not how you spelled arginine. Had all the enzymes needed to synthesize arginine from the precursor molecule. So it was a great control for that reason. And you'll notice that the wild type in all 4 of the growth media and all 4 of the conditions that these Neurospora crassa were grown in, the wild type grew in all the conditions. Okay? So it looks like all the conditions had at least minimal media. That's what MM stands for. We're told that's minimal media. Minimal media only had the precursor molecule present. So minimal media only had the precursor molecule present. But that was no problem for the wild type because the wild type had enzyme A, enzyme B, and enzyme C. Gene A, B, and C were all intact, so it could make enzyme A, B, and C. Enzyme A allowed it to make, again, ornithine from the precursor. Enzyme B allowed it to make citrulline. Enzyme C allowed it to make arginine, and arginine was what it needed to survive. So as long as it had access to the precursor molecule, it was going to grow. If you also gave it access to ornithine or citrulline or arginine, it was also going to grow. This grew with or without any supplement. The mutants were a little bit of a different story. You'll see that the mutants, not all of the mutants grew on all of the growth media. Class 1 mutants, they only grew on these 3 growth media. And it makes sense why they only grew on that growth media when you look at the mutation they had. The mutation that class 1 mutants had was a mutation in enzyme A that made enzyme A non-functional. If enzyme A was not able to function, class 1 mutants could not make enzyme A, could not make a ornithine from a precursor, and so they could not make citrulline from ornithine and arginine from citrulline because they weren't even able to make ornithine. If you gave them ornithine, though, they did still have enzyme B. They were able to convert that to citrulline and then the citrulline to arginine. So they were fine on this growth media. They just were not fine on this minimal growth media. Class 2 mutants, they were fine on the media with citrulline and the media with arginine, but they would not grow if they were only given ornithine or minimal growth media. That's because these class of mutants had a mutation in gene B. Enzyme B was non-functional. They could convert the precursor to ornithine, but once they had ornithine, they could not convert it to citrulline. So even if you gave them ornithine, they wouldn't be able to convert it to citrulline. They had to have citrulline, or you had to straight up give them arginine for them to live. And the last class of mutants was a very similar story in that they had a mutation in gene C. So they only grew on the media that had arginine because they were not able to convert citrulline to arginine, which they needed to survive. So what questions do y'all have there about that experiment? So again, that was just them looking at that connection between genes and proteins, and specifically they were looking at enzymes here. Of course, scientists, we now know that not all proteins are enzymes. We have non-enzyme proteins, and so researchers later revised the one gene, one enzyme hypothesis to the one gene, one protein hypothesis, which then got quickly revised to the one gene, one polypeptide hypothesis, because scientists also realized, oh hey, some proteins are made up of multiple polypeptides, each of which is encoded by its own gene. What type of structure do proteins that are made up of several polypeptides have, y'all? Quaternary, yes, that is quaternary. So there are certain proteins, not all, that have quaternary structure. They are made up of several polypeptides, and each of those polypeptides can have its own gene coding for that polypeptide. It is very common to refer to gene products as proteins, though, instead of polypeptides, so we often see that. Okay. So that's our little gene and protein link, the link between the two. I'm going to, for the sake of time, since we have about 7 minutes left in class, and I want y'all to get some information down so we start kind of where everybody else is, I want y'all to copy down here the basics of transcription and translation. This is just rehashing the central dogma again. We talked about DNA to RNA to protein being the flow of genetic information. We make RNA from DNA via transcription, and we can build a protein using a RNA template via the translation process. So the synthesis of RNA, specifically the synthesis of messenger RNA, or mRNA for short, is what happens during transcription. Translation is the synthesis of a polypeptide, and the cell uses the information in the mRNA in order to synthesize a polypeptide. Ribosomes will read mRNA. They'll read the code of the mRNA, and that is how they know how to build a specific protein. In prokaryotes, translation of mRNA can actually start before transcription has even finished. So in bacteria cells, for instance, the mRNA can be used to start making a protein even before we have finished making that complete mRNA transcript. That is because there is no nuclear envelope to divide the processes of translation and transcription in prokaryotes. In eukaryotic cells, the nuclear envelope is going to separate transcription from translation. The mRNA transcript will undergo some processing before it leaves the nucleus. And that's a little extra layer step of control that eukaryotes have during their process of gene expression. Anyone still using what I have on the board there? Okay. Sorry, y'all. We're going to do this page. We'll do this last page, and then okay. So here, I just wanted to summarize a little bit of what we were talking about on the last page about that transcription translation thing, because that is really important. Here, I'll give you. That is really important for when we start to talk about the differences in gene expression in prokaryotes and eukaryotes. I want y'all to know that transcription and translation take place in the cytoplasm in prokaryotes, okay? There's no membrane-bound organelles in our prokaryotic cells like bacteria and archaea, and so they are going to do transcription and translation both in the cytoplasm. They can actually start using the mRNA to make a protein before that mRNA has even finished being transcribed. That is not the case in eukaryotes. Transcription and translation are clearly separated by a nuclear envelope. So our primary transcript, or our pre-mRNA, undergoes some processing before it leaves the nucleus and is potentially translated. And the triplet the triplet pairs of nucleotides, pairs, they're not pairs because they're triplets, the three nucleotides that help to determine what amino acids get selected for and used to build a polypeptide are what we would refer to as codons. Codons are triplets of nucleotides. They're like words that a ribosome reads in order to build a protein. It's a little instruction manual for building proteins. I'm going to move this down a little bit. Am I good to move this down to here? There's more, I know. We're almost done. This is the last page. And then I'm going to record myself teaching the next page. That way the video is complete for all the other classes. But the rest we will do next class. So, again, those codons, or those triplets of nucleotides, they're like little words that a ribosome reads. The words are translated into a chain of amino acids by the ribosomes to help form a polypeptide. So our flow of information, DNA to mRNA to protein, one of our DNA strands will serve as a template strand. That is going to allow RNA polymerase to come in, make an mRNA transcript, and then during translation, the mRNA codons are read in the 5' to 3' direction by a ribosome. So DNA is replicated in a 5' and 3' direction. mRNA is created in a 5' to 3' direction, and mRNA is read in a 5' to 3' direction. The only difference between RNA and DNA is those base pairs typically, and obviously the sugar as well. But thymine is replaced by uracil and RNA, and each codon specifies the amino acid to be placed at the correct position in a polypeptide. I will finish this video up to page 13 and post it on Canvas. And this class together, we will go over page 13 next class since we didn't get to that. Okay. Thank you all for hanging in there with me. I'm going to pause this and then finish my page 13 here in a second. Okay, so to finish up the video today, we're going to go ahead and flip to page 13. here. I mentioned on the previous page that the codons are what determine which protein is ultimately going to be added to a growing polypeptide chain during the process of translation. I want you to notice the code we have cracked. We know which codons code for which amino acids. We figured that out in the early 1960s. And so the chart here is going to tell you which amino acid is coded for with which codon. The only one you need to memorize is AUG as the start codon. You do not need to memorize this whole chart. We are not expecting you to do that. But you do need to know AUG is the start codon. That one I think is not too bad to remember, though. I always remember that AUG is the start codon because school starts in August. So if you remember, oh, school starts in August, AUG is my start codon because that's the abbreviation for August. One of the things you will notice in the codon chart, though, is that there are several codons that will end up coding for the same amino acid. For example, UUU and UUC both code for phenylalanine. UCU, UCG, UCA, and UCG all code for serine. And so in that sense, we say that the genetic code is redundant, meaning that, again, more than one codon can specify a particular amino acid, like UCU and UCC, both coding for serine. The genetic code, however, is not ambiguous. We say that no codon specifies more than one amino acid. So UCU here is, we're going to say, always codes for serine, or UCC always codes for serine. So genetic code, again, is redundant but not ambiguous. In order for the correct polypeptide to be produced, another thing that has to happen is the codons must be read in the correct reading frame or groupings. So I have some examples of some reading frames down here underneath this diagram as well. So I'm going to go ahead and zoom in on that so you can see. There's a couple of different reading frames here. We have reading frame number 1. If you start reading directly from the beginning, where we start here, AGU, okay, AGU codes for serine. The next three triplets here code for leucine, CUU, okay? So we have serine, leucine, threonine, alanine, so on and so forth as we look at this reading frame. Now, if we shift our reading frame over one base so that instead of looking at AGU as our first codon to code for the first amino acid, and instead we look at this triplet here, GUC, GUC codes for valine. So you can see how just shifting your reading frame over one base can result in the production of a very different polypeptide. We'll talk more about how important reading frames are again when we get into frame shift mutations next class, because the reading frame is super important when it comes to frame shift mutations. Okay. The other thing I wanted to remind us about is that the genetic code is nearly universal. It's shared by the simplest bacteria up to very complex multicellular organisms, and that similarity, that universal genetic code, is evidence for the shared ancestry of all organisms. With the theory of evolution, we do believe in modern biology that all organisms are descended from something called LUCA, the last universal common ancestor. And the fact that all organisms share the same 4 bases in their DNA is some evidence of this shared ancestry. The other interesting thing about the genetic code being universal and the different base pairs being used by all organisms is that genes can be transcribed and translated after being transplanted from one species to another. We're actually going to do this in our transformation lab a little bit later this unit, but we also do this for a lot of different other purposes, like medicinal purposes. The bacteria, for example, we use bacteria to grow human insulin in order to help treat people who have diabetes. If we put the gene for human insulin into bacteria, or what scientists did is they actually kind of engineered a gene for human insulin and then put it into bacteria, they are able to make bacteria create human insulin for us. Prior to us figuring out how to insert a gene for human insulin into a bacteria and get the bacteria to make that human insulin for us, we had to harvest insulin from other sources like cow pancreas or pig pancreas or sheep pancreas. And some people actually had reactions to the insulin from those sources. So the engineered insulin is a little bit better option in terms of treating people who have diabetes and need access to insulin. And the last little thing I want to talk about here today is transcription, and then we'll finish up the rest of transcription next class. The transcription process is facilitated by an enzyme called RNA polymerase, and I did put a star by it because this is one of those enzymes that you do need to know. RNA polymerase specifically is going to make a mRNA transcript using DNA as a template. So it will pry the DNA strands apart, and it will start to lay down some RNA bases in the form of an RNA transcript. That RNA transcript is complementary to the DNA template strand. It's going to use a DNA template strand to figure out what bases to lay down. And just like DNA polymerase, RNA polymerase also elongates in a 5' to 3' direction. It does not need a primer like DNA polymerase, though. RNA synthesis is going to follow the same base pairing rules as DNA, except, like we mentioned earlier today, RNA is going to use uracil instead of thymine. So U pairs with A in RNA, T pairs with A in DNA. And RNA polymerase knows where to start transcribing because there is a special DNA sequence called the promoter region that it can attach to. There's also some things called transcription factors that will facilitate RNA polymerase's attachment to a promoter region, but we will talk more about those later. For right now, I just wanted to mention that RNA polymerase knows where to start because of that promoter sequence, and it knows when to stop transcribing because of a special terminator sequence that ends transcription. The stretch of DNA that is transcribed is referred to as a transcription unit. Our completed RNA transcript in a eukaryotic organism will then undergo some RNA processing. And so we will pick up there next class. Thank you for tuning in. And let me know if you have questions.